Before we get started with this lecture, I'm going to caution you that either at the end of towards the end of this lecture or the beginning of the next, you're going to be missing some slides. So perhaps before you start really listening to this, you might want to jump off, go to Blackboard, and go to the lecture folder. In the Exam 3 folder, in the lecture folder, you're going to find these slides noted as PNS bonus, all right, peripheral nervous system bonus. And those are the slides that you're going to need to complete either the end of this lecture or certainly the next lecture. So you might want to take care of that. So now we're at the cerebrum. The cerebrum is our last region of the brain, and many would say that this is what makes us most quote unquote human. Okay, in psychology classes, this is what they're probably going to spend most of their time on. All right, this is where emotions, thoughts, those sorts of things are going to be generated. And it's felt that this is the quote unquote brain, but as we've seen, this may be where thinking and memory and those sorts of things occur, but without those other three regions, we're not going to be alive. That's what keeps us alive. All of those homeostatic functions, those respiratory centers, those sorts of things, that's what allows us to stay alive, for the cerebrum to do all of those wonderful higher level things that we say it can do. So when we look at the cerebrum, the surface is made up of gray matter, much like the cerebellum. I think of this as a cake with icing on it. Okay, the icing is the gray matter, that's kind of the outer layer. We call that the cerebral cortex. The cake part is the white matter beneath, right? So the cake is going to be the white matter that's going to be deep to the gray matter, which is the surface covering of the cerebrum. The cerebrum is going to be divided into left and right halves or hemispheres. The divider for those hemispheres is the longitudinal fissure, the long, deep crack, if you will, that's going to divide the brain into left and right halves. When we look at the surface of the cerebrum, we're going to see wrinkling, right? Ups and downs, hills and valleys, as I refer to them. First, we can talk about a gyrus. A gyrus is one of these structures. A gyri are several of these structures. These are the folds. These are what I would refer to as the hills. So anytime you see a hill, it's referred to as a gyrus. All of those hills together are the gyri. It is believed that these folds help to increase surface area because there's going to be more neurons making up all of those folds. For a long, long time, it was felt that if somebody was really, really, really smart, they would have a terribly folded, right? There would be tons and tons of gyri, more than average, quote unquote, more than average. So when Albert Einstein, arguably one of the brightest humans that we know of, that we have on record, we got hold of his brain. We thought, oh my gosh, it's going to be wrinkly. There's going to be tons and tons and tons of these gyri. Well, we popped open his head, and guess what? He had an average number of gyri. So although it does increase surface area, there's not any kind of magic connection between the more gyri you have, the smarter you're going to be. All right, so that's not necessarily a documented if-then kind of a statement. So if the gyri are the hills, that means a sulcus will be the valley. And again, a sulcus is one, sulci is many. These are the grooves between the gyri. I've added this diagram to kind of give us an illustration. So the sulcus are the valleys, right? Those two grooves on either side of a gyrus, which is the hill. Now, when you look in your textbook and you're struggling through this stuff with lab, you will notice that virtually every gyrus, virtually every sulcus has a name. For lab purposes, we don't really care. If on your lab exam we're looking at a hill, you're simply going to say that it is a gyrus. If we are looking at a valley, you're simply going to say that it's a sulcus. Although they all have names, we're not going to worry about those, right? That's not anything that we're going to be concerned about. So when we look at our cerebral hemispheres, we're going to divide that up into lobes. We have a frontal lobe, a temporal lobe, occipital, and parietal lobes. Hopefully, this is going to make you say, wait a minute, I know those names. Where do I know those names from? They are named for the bones that lie over top of them. All right, so when we were looking at the anatomy, we saw the bones first and we named the bones. And then when we peeled the bones away, we said, okay, we're going to call this section under the parietal bone the parietal lobe. This section under the frontal lobe, the frontal, this section under the frontal bone, we're going to call that the frontal lobe. Now, what we do know is each of these lobes have specific tasks that they are in charge of. The frontal lobe is in charge of voluntary motor function. Right? So if you're thinking, okay, I'm going to pick up my pen and write something, that signal is going to come from the frontal lobe and be part of the afferent division. The frontal lobe is also very active in motivation, aggression, and mood. So again, we have yet another section of the brain that is going to play into how you're feeling, how your mood is, what your motivation level is. This is where a lot of higher level thought comes from. This is where a lot of planning comes from. And it's one of the slower areas to fully mature in late adolescence. So in your early 20s, this is when the frontal lobe is fully developed. Next, we have the parietal lobe. It's going to be separated from the frontal lobe by a central sulcus. Now, I call the central sulcus to your attention because it's one of the major sulci that you're going to be able to identify. Next slide is going to be a diagram, and I'll point it out to you. In the parietal lobe, we're going to find a center for reception and evaluation of sensory information, excluding smell, hearing, vision. They're going to be taken care of in other lobes. Now, again, I've been trying to drill this into your heads because as humans, we're based on the senses of smell, hearing, and vision. We've got to remember that most of our sensory information has nothing to do with what we would call, the more common senses that we're worried about: smelling, hearing, vision, those sorts of things. These are going to be centers for the reception and evaluation of stretch receptors, pressure receptors that we call baroreceptors, chemoreceptors, those kinds of receptors that are in charge of monitoring oxygen level in the blood, glucose level in the blood, those sorts of things. That sensory information is going to be sent to the parietal lobe and monitored there. So we have our frontal lobe, we have our parietal lobe. Our frontal lobe is kind of in a pink color. Our frontal lobe is in a kind of a pink color. The parietal lobe is kind of in a yellow color. And that central sulcus is going to be a valley that literally connects or runs from the longitudinal fissure down through and kind of ends at the temporal lobe. Right? Because it's such a major sulcus, I do call it by name because it is the divider, the de facto divider between the frontal and parietal lobes. Next is the occipital lobe. This is responsible for the reception and integration of visual input. This is telling you what you're seeing. Right? When you're looking at this screen, at your computer screen, and you can see these words, that information is coming from the occipital lobe. You'll remember that there was a visual reflex center in the brainstem. Okay, and that's just to kind of keep you out of danger. The occipital lobe is interpreting what you see. Right, so it takes a little bit longer, but it's going to give you much more information than simply, holy crap, I need to duck. Then we have our temporal lobe. It receives and evaluates olfactory and auditory input. Olfactory is your sense of smell. Auditory is hearing. It is separated from the rest of the brain by the lateral sulcus. And that's what you're going to see the central sulcus kind of intersects with this lateral sulcus. Gray matter in the cerebrum is located on the outer portion. Again, we call it the cortex, the cerebral cortex. Gray matter forms clusters inside the cerebrum, and again, these are our nuclei. Typically, these nuclei are going to be made up of neuron cell bodies that we're going to find within the central nervous system. When we find a clump of, or a cluster, use the same word, a cluster of neuron cell bodies in the peripheral nervous system, we refer to it as a ganglion, right? But when it's in the central nervous system, it's a nuclei. The white matter, deep inside, the white matter makes up the remainder of the cerebrum. We call that the cerebral medulla. It's organized into nerve tracts that will allow the various parts of the central nervous system to communicate with one another, to allow a signal to be sent from one section, one region of the central nervous system to another. Because again, remember, white matter is going to be made up of myelinated axons, typically, almost always axons, but also dendrites. Right? That's why they're white, because they have that great amount of myelination. Now, when we talk about these tracts, we can identify 3 specific types of tracts. The first is association fibers, and if you try to look at what the name means, it makes it easier to keep track of what they're going to do for you. The association fibers associate areas of the cerebral cortex within the same hemisphere. So association fibers allow all of the sections of the left hemisphere to communicate with each other, or all of the sections of the right hemisphere to communicate with one another. Okay? It's staying in one hemisphere or the other. Next, we have commissural fibers. They're going to connect one cerebral hemisphere to the other. An example of this is the corpus callosum. Years and years ago, when I taught a completely other course, we would do a pig dissection. In the last week of the pig dissection, we would open up the skull and get the brain out. And one of the really cool things for students was that you would be able to gently pull apart or separate the 2 hemispheres, because that longitudinal fissure is in fact the divider between those hemispheres, and there's no connection between the 2 hemispheres until you get to the corpus callosum. The corpus callosum is a bundle of neuron fibers that allows communication between the left and the right hemispheres. And last, we have projection fibers. They connect the cerebrum with other parts of the brain and the central nervous system, so they're going to project the information outside of the cerebrum. Now, you may have heard of the corpus callosum in a psychology class, because to help minimize really, really severe epileptic seizures, we can, it's a last resort, sever the corpus callosum. What an epileptic seizure results from an electrical impulse, because again, all of these neuron impulses are electrical currents just kind of ricocheting around the brain. And if we sever the corpus callosum, that minimizes how much of the brain is going to be affected by a seizure. It doesn't stop the seizure, but instead of allowing it to go from the left hemisphere to the right hemisphere and back again, we sever that connection, and it's going to remain in the hemisphere where it starts. This is a coronal section of your brain. You can see right in the upper left corner how we're cutting it. And more than anything, I want to point out to you the corpus callosum. It's these green kind of strandy guys. That's the connection between the left and the right hemispheres that allow those hemispheres to communicate with one another. Next is the peripheral nervous system, because we talked about the central nervous system, the brain and the spinal cord. Next, we're going to start our peripheral nervous system, and this is where you're going to need those quote-unquote PNS bonus slides. So you're going to want to get those